Hello, my name is Sohn, and today I'll present on the effect of epigallocatechin 3 gallate on inhibiting beta amyloid aggregation in Alzheimer's disease. It's the most common form of dementia, and it was the seventh leading cause of death in 2021 despite COVID. According to the Alzheimer's Disease International, 46 million people in 2015 had dementia, and this number is predicted to increase by 131.5 million by 2050. In terms of pathogenesis, it is very unclear the specific mechanisms behind it, but we do know that it is due to the accumulation of amyloid plaques and neurofibrillary tangles. In excessive amounts, this buildup causes abnormal excitation, decreased activity, and death in neurons. Subsequently, we can observe behavioral changes like cognitive decline and memory loss in patients. Its neurofibrillary tangles are less specific to AD and are seen in a greater range of dementias. Beta amyloid itself is a normal peptide primarily produced by cleaved amyloid precursor proteins, while in its excess aggregation in the form of amyloid plaques is the most distinctive neuropathological hallmark of AD. As you can see in the photo to the right, beta amyloid monomers are able to aggregate into various levels of assemblies from monomers, dimers, oligomers, protofibrils, and amyloid fibrils. Simpler assemblies of beta amyloids, like oligomers, are smaller and soluble, and they are spread throughout the brain. However, complex assemblies of beta amyloids, like amyloid fibrils, are larger and soluble and therefore form neurotoxic fibrils. Recent studies suggest that Alzheimer's progression is driven more by the type and origin of beta amyloids than by plaque accumulation alone, with certain APP mutations and non-APP mutations causing synaptic damage even in the absence of plaques. This challenges the traditional view that amyloid plaques are the primary cause of this disease and therefore draws more importance to the study of beta amyloids in itself. Epigallocatechin 3 gallate, or EGCG, is the most abundant catechin found in green tea, and it's thought that these polyphenols, though their individual mechanisms are unclear, they are responsible for the health benefits such as antioxidant, anti-inflammatory, and anti-neurodegenerative properties. Various studies have shown their inhibitory effects, reducing overall loads of amyloid deposits, decreasing amyloid aggregates, and lowering beta amyloid aggregation. The mechanism of such is not fully understood, but some studies suggest that EGCG directly binds to beta sheet fibrils, that is, beta amyloid aggregations, destabilizing them and preventing further assembly. They have also been shown to prevent secondary nucleation, where new beta amyloids aggregates form, the binding to fibril ends and preventing monomer aggregation. They also redirect beta amyloids from forming beta sheet fibrils into forming non-toxic aggregates with minimal beta sheets. This makes aggregates more soluble and easier to break apart. And they also enhance the activity of neuronal immune cells, allowing them to break down further complex assemblies of beta amyloids. Main goals of this study. The first is to measure the effect of EGCG on beta amyloid aggregation. This is through quantifying the effect on aggregate area and size and finding whether or not it has an inducing or inhibiting effect on aggregation. Second, it is to identify an optimal EGCG concentration. So this is at which concentration beta amyloid aggregation is at a minimum, therefore inhibiting its aggregation. Third, it is to identify recommendations for clinical applications and how we as individuals can apply our knowledge of EGCG on AD in real life, in terms of their dosage, delivery, and value in research. Utilized methods such as the thioflavin T fluorescence assay or light spectrophotometry. This experiment instead quantified aggregation to the total area of beta amyloid aggregation because of limitations of lab equipment and materials. To prepare samples, I first made these tubes by diluting a stock EGCG concentration to my fixed IV intervals of 0, 10, 25, 50, and 75. I then incubated these samples at 37 degrees Celsius for 2 days, and I observed them on each day. To observe them, I stained each sample with 0.01% of Congo red before being observed. Of course, they were also let to rest as well. These photos were taken, there were 3 photos taken per sample to have 3 repeats per sample. Data, I used 4 main techniques. First, I used ImageJ, which is an open source imaging software, to manually select aggregate areas in pixels and convert them to nanometers. I did this per category of aggregate, which was small, medium, and large, which was determined by scientific knowledge and will be explained later on. After these data sets, I averaged them out and for per repeats to form trial averages and then a total average per aggregate size. I calculated this per category, and I also calculated standard deviation per each one of these data values. Then, in order to understand the significance of my data, I performed a one-way ANOVA test, which attempted to find the significance of neuroconcentrations with a p-value of 0.05. finally, I performed a 2-key HSD test, which found which concentrations were above the critical Q value and thus made a significant difference. Move on to quantitative data, I'd like to first discuss a little bit of qualitative data. So as you can see, in the first row is my results on day 1, and second is my results on day 2. I took these randomly and from one fixed interval. From here, you can see that there was little to no aggregation and the zero concentration, and it peaked at 25 and then exponentially decreased at 50 and 75. You can see you can see how clearly it is where the opaque little black molecules are the beta amyloids, while you can see sometimes it's very hard to see, but the somewhat translucent one are the EGCG particles. Aforementioned, I first used IMageJ, which is an open source image processing program to analyze beta amyloid aggregation. Due to variability in image brightness and visual noise, given the method of measurement, I was not able to threshold and yield reliable results. Therefore, aggregate areas were manually selected using the wand tool to ensure accurate and consistent measurement. Given beta amyloids were very clear in the previous image, you can see, relative to other particles, a fixed selection of 40 tolerance was kept for selection, and prior to selection, images were enhanced with 8-bit sharpening, enhancing contrast by 0.35%. Measurements were taken in pixels by IMageJ and later converted to square nanometers using stage micrometer calibration on Excel. Thus, you can see this white photo over here with the white background is my raw data, and I later processed to form this final photo. To categorize the process of successive aggregation, aggregates in each photo were grouped into 3 categories: small aggregates, which is below 20 nanometers, intermediate aggregates, which was in between 20 nanometers and 200 nanometers, and large aggregates, which was above 200 nanometers. This was in line with scientific research, and this is to reflect the different levels of beta amyloid assembly: early oligomeric species, growing protofibrils, and mature fibrils. This data was then organized in Excel for each picture and trial and EGCG concentration. EGCG has a concentration-dependent effect on beta amyloid aggregation, with the extent of this aggregation varying distinctly across different aggregate sizes. At 0 micromolars, the average aggregation is relatively low, and at 25, it peaks. Once again, it suggests that lower concentrations may unexpectedly enhance aggregation rather than inhibiting it. We see that after 50 micromolars, this significantly decreases of beta amyloid aggregation, and at 75, this actually plateaus, where we kind of note that there was a small increase in the aggregation of large aggregates, while the medium aggregates decreased slightly. This suggests that perhaps the optimal concentration is indeed 50 micromolars, preventing further beta amyloid aggregation. However, one main concern is the high standard deviations observed in the data, particularly at 25 micromolars, with significant variability in beta amyloid aggregation. This is particularly attributed to the nature of beta amyloid aggregation and the small concentration of beta amyloids I had due to the cost of beta amyloids, but the overlap in error bars do significantly impact the reliability of the data throughout the experiment. One way ANOVA is to measure to determine whether there are significant differences between the means of independent groups. In this experiment, it was used to assess whether or not different concentrations of EGCG resulted in significantly different levels of beta amyloid aggregation. A low p-value, that is 1 under 0.05, would indicate that EGCG has a measurable impact on aggregation, while a high p-value would indicate the difference is more attributed to random chance. Therefore, a low value accepts the alternative hypothesis, while the high p-value would accept the null hypothesis. And the results from this table show highly significant p-values for all aggregate sizes, which means that they are below the threshold of 0.05. This indicates that, indeed, changes in EGCG concentration lead to statistically significant differences in beta amyloid aggregation across all measured size categories, rejecting the null hypothesis and accepting the alternative. The smallest p-value is seen for total aggregation, suggesting the most significant overall impact of EGCG on beta amyloid aggregation. Test is used after a one-way ANOVA test to find out which specific group means differ significantly. It calculates the difference between each pair of groups means and standardizes it using the within group variance. Each standardized difference, aka a Q value, is then compared to the critical value based on the number of groups and degrees of freedom. The degrees of freedom provided by the 2-kit HSD test, where the number of groups was 5 given my IV intervals. If the Q value is greater than or equal to the critical value, the difference between those groups is considered statistically significant. So in this image, or in the slide, you can see to the left is an example of one of the tests I performed for total aggregation. In the second column, you see the absolute difference, the third standard error, and therefore I found my Q value, and I compared to the critical Q value to find whether or not there was a significant result. Specifically for total aggregation, I found that it was more stark, and here you can see I noted red as in not significant and green as in significant. It's kind of hard to see with the background, but you can kind of see. To the right is my summary of my results, which show the overall results of the Tukey HSC test. As you can see from these results, there were slight variations in results of small, medium, and large aggregates where results could have been significant and were very close to, but were not ultimately significant. However, in total aggregation, these results were very stark, and it was either significant or not significant. The most significant out of every single group, unanimously, was the 0 versus 25 concentration groups, showing that indeed aggregation does occur exponentially, and 25 micromolars perhaps could induce beta-amyloid aggregation. Significance also found in 25 versus 50 and 25 versus 75 concentrations, showing and supporting that indeed concentrations of 50 and above are optimal for inhibiting beta-amyloid aggregation. Levels of EGCG at 50 micromolars do obstruct further beta-amyloid assembly, both in larger aggregates and abundance. At lower concentrations, it seems that while there are fewer large aggregates, there is still abundance of beta-amyloids, which is known to be neurotoxic, and perhaps they have an inducing effect on beta-amyloid aggregation in itself, which is similar in line with scientific research. Higher concentrations seem to have a minimal effect on large aggregates, while decreasing the overall amounts of intermediate aggregates. Despite this, EGCG still presents itself as an effective inhibitor of amyloid plaques and thus impeding AD progression and potentially treating later stages of AD. However, a distinct lack of beta-amyloid preconditioning at a concentration of 0 micromolars fails to establish a baseline assessment and therefore a line of comparison. And though the intervals do suggest efficacy of 50 micromolars, a baseline assessment would be really helpful to compare the proportion of which ECG is able to deaggregate beta amyloids. Additionally, further studies with longer incubation periods, fluorescent markers, and further imaging techniques could enhance detection of early aggregation and thus its inhibition. In clinical application, further consideration is needed for the absorption of EGCG given the permeability of the blood-brain barrier, otherwise known as BBB. Existing studies have shown the ability of EGCG to be absorbed by the BBB, proving EGCG to be a more effective and non-invasive method than other therapeutic approaches. Obviously, we are not encouraging participants inject green tea into their veins, but more as if this is one out of many ways in which patients could potentially reduce their risk or inhibit their progression of AD.